Thank you everyone for standing by and welcome to our webinar entitled Turning Dredged Sediment into Farm Soil Nutrient Supplements. This webinar series called Freshwater Science will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal bloom and human decision-making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Angelica Vasquez Ortega at Bowling Green State University. Dr. Vasquez Ortega is an assistant professor of geochemistry in the School of Earth, Environment, and Society at Bowling Green State University. Her research focuses on nutrient and carbon distribution in agricultural soils, the parameters of phosphate removal from agricultural tile drainage, and the effects of lake sediment on crop yields. She holds a master's and PhD degrees in soil, water, and environmental sciences from the University of Arizona, and a bachelor's in environmental science from the University of Puerto Rico. We're delighted to have her here today to talk about her research. But before we do that, a few things about the webinar itself. During our presentation, all participants are in a listen-only mode. Afterwards, at around 12:20, I'll conduct a question and answer session. If you'd like to ask a question during the presentation, please feel free to pull up that chat feature anytime during her talk, and I will collect and pose your questions out to Dr. Vasquez Ortega at the end of her presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we'll post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes to fill out that survey. It will help us continue to bring you better webinars. So without any further delay, I'd like to introduce Dr. Vasquez Ortega, presenting Turning Dredged Sediments into Farm Soil Nutrient Supplements. And I think you are ready.
Thank you so much, Jill, for the introduction. I think that then I will jump in directly into the meat of the presentation. Again, thank you everybody for joining this webinar today. We are about 92 participants, so that's pretty nice to see. Okay, again, today's presentation is going to be regarding about my research at Bowling Green State University during my tenure track period. I have been at BGSU for almost 6 years, so going for tenure as we speak. Okay, I will show you a little bit of what I have been doing regarding using dredged sediments as farm soil amendment. A little bit of background information for those that are not very familiarized with dredged sediments. Here, this map I'm showing, let me use maybe the marker, okay, the laser. So here, this figure here is showing you guys the federal navigational channels within the Toledo Harbor. And each year, these federal navigational channels are being dredged in order to accommodate different ships or boats that are bringing different goods to the area. So the accumulation of sediment, it's so that this port have to be dredged yearly. There are other ports that are only maybe dredged every 2, 3, 5 years, but Toledo Harbor is dredged yearly due to the high sedimentation process. So typically the way that this has been done is all the sediments are dredged, they are put in a barge, and then this red square that you guys see here, it's an area that has been dedicated for open lake disposal or placement of the dredged sediments. But the state of Ohio, it's very aware and worried regarding that the open dredge disposal might be exacerbating algae blooms. And for that reason, now we can also, you guys can also see this this island here that is anthropogenic of nature, in which from now on all the sediment are going to be placed in this area. And this will result in a huge amount of sediments that will need to be used for whatever beneficial uses. And the beneficial use that I am proposing is So since there's no more open dredge disposal, let's bring back the soil that was eroded from our very fertile farmland. So this table here, it's showing different parameters regarding the dredge material based on a characterization versus optimal values that a farmer will look farmers will look in their farm fields. For instance, here pH between 5.3 to 7. The dredge material has a pH of 7.9. All right, if we look by the bioavailable phosphorus by the Bray method, farmers are looking between 15 to 40, and the dredge can provide 40 to 80. Also, you guys can observe that the dredge sediments are quite rich in calcium. So these are more of macronutrients that are beneficial for the plants. But the following table, I want you guys to focus more on the lower portion. And here I have, again, different parameters and values for a typical farm soil. It's a Hoytville soil in the area, and then those values for the dredge material. Because one of the questions that I'm being asked constantly is, how contaminated are these sediments? And do we have to worry about that contamination if we put the dredge sediments in farmland to grow different crops, either row crops or specialty crops? So let's, for instance, look at a couple of the different potential heavy metals, like for instance, chromium here. The background value in terms of parts per million or milligrams per kilogram of soil, it's 80 in the farm soil, and for the dredge, it was 80 as well. Copper, cobalt, also 11, 12, nickel, 30, 40, copper in this case, 40, 30, zinc. So you guys can see that actually the dredge sediment concentration regarding these background metals, it's pretty much the same or very similar to those in a typical Hoytville farm soil. So based on this analysis, the Ohio EPA and myself, we have concluded, at least with this data set, that the dredge sediment, it's not posing any detrimental effect if we put it in the land, because again, the values are very similar to any typical farmland. And even though the values look like that, I'm also looking in my research at the bioaccumulation of different heavy metals in the biomass. So even though they are, again, background concentrations, we're also looking at the bioaccumulation once we harvest the different crops. So here, what I want to show you, it's a couple of examples of some greenhouse experiments that I have conducted with my students. Of course, nothing here wouldn't be possible without my students. So in this specific one, we had in the greenhouse buckets growing soybean. And I did a series of different ratios regarding soil and dredge material. Here I have 100% soil, 10 dredge, 90 soil, 20 dredge, 80 soil, and 100% dredge. And so again, the idea is that I have two end members, 100% dredge and 100% farm, with some sort of mixtures in between, because a farmer will not be farming in a huge amount of dredge. It will be amending the soil by some sort of ratio, any acres per, any tons per acres, for example. So the data that I want to show you here is, again, in the x-axis, I have all the different treatments, and in the y, I have different values. In this specific one, I have soil yield, and in the other one, I have root biomass. And what I want to show you guys is that the average of the soybean yield, it increased slightly as the dredge sediments, the amount of dredge sediment was increasing. There was no statistical differences among all treatments, but at least we did not observe any detrimental effect on the yield. However, for the biomass, we could observe a relative increment as dredge sediments were added to the system. And I actually have a really nice picture here showing you guys how robust was the root system of the soybean as a function of adding dredge. So we can see here 100% farm soil versus how adding more and more dredge was affecting the bulkiness of the root system. So again, this experiment was for soybeans. I also have, my students have also conducted experiments with corn. In this case, also looking at different ratios, and I'm gonna show you the data that we have collected. So here, again, I have my different treatments, different parameters, corn yield in this case, and in overall, we can see that the more dredge, the higher the corn yield. And the root system, we also observe a similar increase as more dredge sediment was added. And I'm accompanying that with, again, real pictures so that you guys can appreciate how how robust and bulky was the root system for corn when dredge sediments were included in the mixture. Another greenhouse that my students have conducted, it's more using specialty crops. In this case, we did the experiment with lettuce, tomatoes, and carrots. Again, the two extremes and a 10-90 dredge ratio. Here I'm showing you guys different images of how the plants look in the greenhouse setup. So here we have the tomatoes, the lettuce, and the carrots, accompanied with how the root system for each crop look like. And again, very strong increase in biomass for the root system. It was observed for tomatoes. It was also observed for lettuce. And in overall, the carrots were slightly longer and more have higher mass than, you know, the one that have 100% dredge than the one that was only 100% soil. So those has been all the different greenhouse projects that my students have conducted. And now the next step will be to move this into a real farm demonstration project. And I got a grant in which I'm working in with the Agricultural Incubation Foundation, which is North Bowling Green. And I rented some organic farm in the area, and I implemented what I have been learning in the greenhouse, but now in an actual field. So this experiment consisted of having a conventional field. Again, it was managed as the farmer will do, and these are organic soils or fields. And then we added different ratios of dredge to the land. One treatment consisted of 40 tons of dredge sediments per acre, and corn was planted. The other was 80 tons of dredge sediments per acre, again with corn. And then we had an additional one with 64 tons per acre, but in this case, we had soybeans. And here I have a series of images to show you guys how the whole process, you know, we moved through the whole process. So all of the dredge sediment was excavated and hauled into the land. Then using a manure spreader, we were able to broadcast the material on top of the different fields. Here it's an example of, if I had a video, but I don't have it in this one, but an example of how it was broadcasted. And then the farmer worked the soil and prepared the land for planting the different crops, either corn or soybeans. Here it's how the fields look like, at least the corn fields look like. And this picture was taken in September, the 2nd of September, then later in October, the fields were harvested. And what I want to show you is some of the information that I have collected so far from this demonstration farm project. So here again, this is the Agricultural Incubation Foundation, and all of these plots are either managed conventionally or organically. These are the fields that I was able to rent, and all of these fields were managed organically. So no synthetic fertilizers or pesticides were placed in these fields, only the dredged sediment in the respective fields was added. And the concentration of, again, heavy metals or other contaminants are relatively low or very similar to background values, that these farms were not, we were able to keep the farms organically. So the addition of adding the dredge did not remove the certification of the land to be organic. Okay, so what I want to show you here, I want you guys to concentrate, let's say, in the field, in the cornfields. They have cornfield 1, 2, and 3. Cornfield number 2 received non amount of dredged sediments. Field 3 received 40, and field 1 received 80. The calculation regarding how much dredge each field was going to be receiving, it has to do with the P concentration, the phosphorus concentration by the Matlick method. So we did a calculation of how much more tons we need to add to the soil to increase the P concentration, but no more than 40 parts per million, which is the tri-state recommended value. So when we did that, and if you guys now, I wanna call your attention to the yellow words here. What I wanna show you is the yields of corn in each field. So again, the field that has 0 tons had 122 bushels per acre of corn. The one that has 40 had 118, and the field that has 80 had 102. So we can observe that the addition of dredge actually decreased the yields, at least for corn. And this is not as we have been observing in the greenhouse. So we need to figure out what's going on in the field that is different to the greenhouse experiments. When I had a conversation with the farmer, here are some of the points that he brought to my attention regarding what might be happening. So he remembered that when he was preparing the land for planting, he had to work the land more. He observed that the soil was more compacted, and this might be due to many passes of heavy equipment broadcasting the material. The material was also broadcasted in May, so the soil was wet and more susceptible for compaction. The farmer has advised me that if we are gonna do this type of experiment again, we should do it in fall, that the soils are not gonna be that wet, and then the compaction will be minimized. The farmer also observed that the fields that received dredged sediment, they contain or they had more invasive weeds. So maybe that also can be affecting the germination of the corn and lowering the yields. But again, when I show this data to different researchers, they ask me, are these values significantly different? 122 versus 118 versus 102. And we only have 1 year data set. So we are trying to see if we can have year 2 to see if maybe it was compaction, maybe it was just a bad year for different, something else in the weather that might be affecting this. But year 2, we're not going to be adding more dredge. We just want to know, after the farmer has worked it out, if the compaction still affect, or maybe the yields are more similar among the fields. If you guys observe here for field 1 for the soybean, this one received 64 tons per acre, and the yield was 52. And in the top, I have other fields that did not receive dredge, and the yields are 58, 45. So quite similar in yields. So we did not observe the decrease in yield for soybean, only for corn. So that's why we need to have year number 2. to see if we can observe different results. Okay, moving on. However, what I want to show you here is what was the effect on the phosphorus, bioavailable phosphorus in the soil. So the x-axis have the different rates of dredge per acre. So here, for instance, 0, 40, 80, all of these with corn, and then 64 for the soybean. The blue is representing before planting, and the orange is after harvest. And what we can observe is that for the field that had no dredge application, once after harvesting, there was a decrease in phosphorus concentration. However, for the fields that received dredge, there was an increase in all of them. Some of them might not be significant difference. We have to do the statistics for that. So here is the net change in P. So for the treatment with no dredge, there was a loss. 16% of the bioavailable P was lost regarding it was now bioaccumulated in the plants. However, for treatments including 40 and 80 tons, the change in P was higher. The system acquired. There was a higher concentration of P, 30% for the 40 and 2 times that for the 80. So even though we observe a decline in yield, potentially due to compaction, we are in the bioavailable P in the soil, it's being enriched. So the application of dredged sediment can add nutrient to the farm soil. So what I want to move for the last portion of today's presentation is to a current project that I just got it. It's out of the oven. And I'm actually looking for a farmer that might be willing to collaborate with me and my research team on this project. So HABRI, we obtained a grant via HABRI, via Ohio State grant as well. And in this case, we're replicating pretty much the demonstration farm that we already had. But in addition to that, we are going to be looking at the loss of P in the surface water and in the subsurface water. So we're going to be adding an echo field to the experiment. And of course, we now have funding for 2 years, potentially a no-cost extension for an extra year. So a little bit more information regarding the echo field. Many of you might be already familiarized on the use of weir for the tile drainage to intercept the flow there, and also H flumes to intercept the surface flow. And both of these systems are going to be accompanied by an ISCO sampler to gather water samples to measure the concentration. And then with the discharge, we can actually have the loads of P that are being lost into the waterways. Right now, we are looking for different fields that can help us accommodate our design, experimental design. So we're looking for about 3 plots of about 3 acres. And we want to make sure that when we evaluate the P level, it's below 40 parts per million, ideally, I don't know, around 25, 30, so that when we add the dredge, we can increase that, but not exceed the 40 parts per million. We are looking for fields that have similar texture, that are hopefully are adjacent, that has a similar slope. And we also want to make sure that we can separate the fields in terms of the hydrology, because we want to make sure that we can only collect the tile drainage of the surface water for the specific area. So if you have a field that might have these characteristics, please contact me. Again, this is looking at that. Regarding the surface area, we want to make sure that when we are installing the instrumentation, we are collecting only surface water for that specific area. And the same with the subsurface. Ideally, if there is an open tile drainage like this one, in many instances, we think that we might have to dig down and actually tap into the main tile drainage system. And that way we can either measure the water flow or the collect water for concentration. So again, if the farmer has organic farm, we're gonna follow that recommendation. If it's conventional, we're gonna be following that. But for the fields that are receiving the dredge, there's no addition of synthetic fertilizer. The fertilizer will be the dredge sediment itself. That will be the amendment. The grant has money for all of this instrumentation. We are planning to have solar panels to be able to operate the system. And so I just want to finalize this presentation with the following questions for the audience. And I know that the audience also have other additional questions that I will be happy to answer. But one of the questions that I have on my research team has that we want to engage and learn more is what concerns come to mind when thinking and implementing a project like this one. So what it's something that worries you or any type of concern. And then what other metrics would you look besides production when implementing a project like this one? And I think that maybe this might be in tandem with looking at the bioavailability of phosphorus, because again, for the example that I show you, we saw a decrease in yield, but an increase in the bioavailable peak. So I want to know from farmers or other stakeholders that might be in this call, what will be the answer for these questions. And finally, I want to say thank you to all the different sponsor agencies that have allow me to conduct all of these experiments. And of course, thank you to my master's students and undergraduate students for working with me in this project. So I'm done, Jill. Please take over.
All right. Well, we have gotten some really great questions during the presentation. So let me get started and ask Dr. Vazquez Ortega as many as we can. And what questions she can't answer today, we'll post later on the website with her answers. So we got quite a few questions dealing with other contaminants that people were asking about, like pesticides and PCBs and other those kind of contaminants. And what they were asking was, were any of those organic contaminants possibly reviewed before the dredge material went onto the soil?
Sure, thank you. So all of the dredge sediments from the Toledo Harbor are every so often, I don't remember exactly how often, and if Vanessa from the Ohio EPA is here, she can help me answer this question better. Every so often, the Army Corps of Engineers, they take sample from the dredge while the dredging activity is happening. And the Ohio EPA send those samples to an external laboratory that they do a different series of analysis. Pesticides are included, also PCBs, PAHs, and heavy metals. In the case of my research, I have measured PCBs. In the data that I have so far for the PCBs is in a Hoytville conventional farm versus dredge. So in that specific one, there was no PCBs in the farm field, and there was no PCBs in the dredge sediment. At least the Arochlor, the different ones that are recommended by the Ohio EPA or the US EPA, they were below detection limits. So in terms of PCBs, there was no PCBs detected in the dredge sediments. And I also look at PCBs bioaccumulation in the soybeans, and there was nothing detected there. PAHs, there was PAHs values in both, in the conventional form as well as in the dredge sediments. And my students are looking at potential bioaccumulation, but so far, all the data that I have seen there, at least with the soybean, remember that I show you many different crops. So far with the soybean, there was no bioaccumulation of PCBs. And for the heavy metals, there was the normal accumulation that happens even when you have an organic farm soil or a conventional soil. So far, they have been very similar. But again, I have my master's students working on this, conducting a statistical analysis, and I will be able to give you more specific information once I have those reports ready to share with different stakeholders.
All right, thank you. We got several questions dealing with how, if you have been tracking or will be tracking microbial communities before and after the sediments go on to the agricultural fields.
Yeah, I have a peer here, Bowling Green. Her name is Dr. Jiawei Xu, and she is working with me, collaborating with me in this project. And she is looking at the abundancy of the different of the microbial community, looking at the DNA. So far we have data of which organisms, which microorganisms are there. We are contemplating doing more about what are they active, looking more at the enzymes. But I also know that there is a professor at Rice State University, Megan Rua. She is also doing this type of research. So I believe that she's looking more not only of what is there, but also how active are these are the microorganisms within the dredged sediments. So again, Megan Rua from Rice State University, she might be a good person to contact regarding microbial activity.
All right. Another question that we had gotten was, how do you explain the differences between field and greenhouse results?
That's a very open question. If the person is referring regarding the yields, I want to know more by adding more additional years to looking at the data and looking at yields over time. Again, the way that the buckets were done, compaction was completely minimized. The student did not compacted any of the material within the bucket. So the root system was able to grow very robust. And in the farm field, we had different results regarding potential compaction. So that's why I want to know if year 2, and then with the new project, we're learning. Now we are going to be, for the new project, we're going to be putting into practice all the different advice that the farmer has been giving me so far. And we are going to be looking at yield. We're going to be looking at soil health, and we're going to be looking at microbial community. We're going to be looking at the safety, the food safety regarding the bioaccumulation of the different organic and inorganic contaminants. But I have all of this data is in the making, so I will know more as my master's students complete their thesis.
All right. We had a question dealing with, are you concerned about invasive species that may be in the sediments, that there might be a risk to transfer those to farmlands?
Yeah, that's a really good question. And I think that it was evident for the demonstration farm field that I was working with, in which it was an organic farm soil, and the farmer couldn't add any pesticides, and he observed the potentiality of, the intrusion of these invasive species. It might be different for a conventional farmer that can treat with different pesticides their land, but it's something that a farmer needs to be aware that it's a potential, negative effect, if you want to think about it, by adding invasive plants. Because the Toledo Harbor is the one that is managing the confined facilities, and these facilities are not managed regarding managing invasive plants. So whatever it's in the air and gets deposited in the sediments, it will grow.
All right. Thank you. We've gotten quite a few questions dealing with the root biomass. And one question specifically, have you considered looking at the differences in the yield to root system when looked at dewatered sediments placed by earthwork equipment versus dredged sediments placed via hydrologically dredging at the farm site? Curious how much the dewatering affects the analysis.
Okay. That was a really long question.
I know, I'm sorry.
No, don't worry. So as today, the dredged sediments in the confined facility, they are placed hydraulically. So it's pretty much 80 to 90% water, and then slowly being the water. The idea at the moment is to excavate and haul the material to the farm field and broadcast it. It will only make sense for farms that are going to be very close by to the confined facility, because it will be cost prohibited the farther you go from the facility. There are no experiments conducted regarding planting the crops in the very same confined facility to see how the difference between hydraulically placed and the water in situ versus excavating and broadcasting in another area. I know that there have been, I believe, other demonstration farms, I don't know if in Ohio and other states, that they have applied the dredge with, all the water that contains, and then it dewaters in the field. I know that the Ohio EPA has contemplated that idea, but those logistics are not my area of research, so I cannot answer that in a specific, but to my knowledge, at least I believe that this project is the only one in Ohio that has been able to broadcast dredged sediment from the Toledo Harbor, and these are pioneering data, so I don't know specifics growing crops in the confined facility in situ.
All right. I have one more question, and then what I'm going to do is I'm going to collect all the rest of the questions and send them over to Dr. Vasquez Ortega's and see if she can answer all those, and we'll post those later on to the site. Here's my last question. Sorry, that was a little long. Will the dredged material only be applied on live plants, or residue from after harvest?
Okay, that's a really good question. And I think that that's something that we are still learning and working with farmers. The farmer that worked with me at the organic farm, he recommended the following strategy next time that I put the dredge. So he would like to have a cover crop. In the one that he uses the more frequently, it's clover. So he would like to have clover cover crop, put the dredge sediments, and then ward the cover crop and the dredge into the system. So that both, and do this in fall, so that both doing this in fall plus the cover crop, it will try to minimize the compaction of the soil. So that's what we are what we are aiming to do in the second project, follow this advice. So as today, we are recommending to broadcast the dredge material on top of a cover crop and then ward the land for planting on May, the next season, the next year, the next spring.
All right. Well, I will close with that question. I wanted to thank again Dr. Vazquez Ortega for her willingness to talk to us today about her research. It was really an excellent discussion. Also, a thank you to Christina Diakis for her work organizing this webinar series. I would like to mention to all of you to look and see that survey URL for the webinar. It's in the chat feature. If you could take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue next month with Dr. Steve Lyons, who will be discussing the impacts of tile drainage to water flow. The registration link is in the chat. Thank you again to Dr. Vazquez Ortega. Thank you so much. This was so interesting, and all the participants in this webinar. We really hope that this was beneficial and hope you'll join us again in an upcoming webinar. Thank you again and have a great afternoon. Thank you again for the presentation. This was great.
Thank you, Jill. Bye-bye.
Thank you.